Hello students, welcome to lecture 43, the last class of 5th module. We had discussed the various gene expression related matters of prokaryotes and eukaryotes with this module. So in this class we are going to discuss the last topics: gene expression, transposition and gene prediction approaches. We will see the overview of this topic in this class. Gene expression is the process by which information from a gene is used in the synthesis of a functional gene product. These products are often proteins, but in non-protein coding genes such as transfer RNA tRNA or small nuclear RNA snRNA genes, the product is functional RNA. The process of gene expression is used by all known life. Several steps in the gene expression process may be modulated, including the transcription, RNA splicing, translation, and post-translational modification of a protein. Gene regulation gives the cell control over structure and function. In genetics, gene expression is the most fundamental level at which the genotype gives rise to the phenotype, that is, observable trait. The genetic code stored in DNA is interpreted by gene expression, and the properties of the expression give rise to the organism's phenotype. Such phenotypes are often expressed by the synthesis of proteins that control the organism's shape or that act as enzymes catalyzing specific metabolic pathways characterizing the organism. Regulation of gene expression is thus critical to the organism's development. Now the major mechanisms of gene expression involves transcription, RNA processing, RNA export, translation, folding, translocation and protein transport. We know transcription means the conversion of DNA to RNA. Then RNA processing, that is splicing. Splicing means the process of excising internal sequences of eukaryotic hnRNAs introns and rejoining of exons that flank them. Then non-coding RNA maturation. A non-coding RNA, ncRNA, is a functional RNA molecule that is transcribed from DNA but not translated into proteins. Abundant and functionally important types of non-coding RNAs include transfer RNAs, tRNAs, and ribosomal RNAs, rRNAs, as well as small RNAs such as microRNAs, siRNAs, piRNAs, snRNAs, snRNAs, xRNAs, scRNAs, and the long ncRNAs such as cyst and hotire. Non-coding RNAs contribute to diseases including cancer and Alzheimer's. Then RNA export. In eukaryotes, most mature RNA must be exported to the cytoplasm from the nucleus. While some RNAs function in the nucleus, many RNAs are transported through the nuclear pores and into the cytosol. Notably, this includes all RNA types involved in protein synthesis. In some cases, RNAs are additionally transported to a specific part of the cytoplasm, such as a synapse. They are then towed by motor proteins that bind through linker proteins to specific sequences. Then translation. We know translation is the process of converting the information from the nucleotide sequence in RNA to the amino acid sequences that make a protein. Then folding, it is often used synonymously with the term structural motif, but typically used to connote large regions of similar secondary structure found in two or more proteins. Then the next mechanism of this gene expression involve translocation, that is, proteins of eukaryotes or prokaryotes must be translocated to enter to appropriate pathways. Finally, protein transports happens via the Golgi apparatus. When we learn gene expression, we should remember some useful DNA sequence features for eukaryotic gene recognition algorithms that we discussed. That is, non-promoter elements like TATA and CAAT boxes, CPG islands, splicing signals associated with introns, open reading frames with characteristic codon utilization, then similarity to the sequence of EST, that is expressed sequence tag, or genes from other organisms. We have an idea about the first 4 DNA sequence features. We have to understand some more details about expressed sequence tags, so that we will discuss in the next slide. So let us understand what is cDNA and EST. In genetics, complementary DNA, cDNA, is DNA synthesized from a single stranded RNA template, that is, for example, messenger RNA or microRNA. So from a single stranded RNA template in a reaction catalyzed by the enzyme reverse transcriptase. cDNA is often used to clone eukaryotic genes in prokaryotes. cDNA, that is complementary DNA, represents convenient ways of isolating and manipulating those portions of a eukaryotic genome that are transcribed by RNA polymerase II. The resulting double-stranded DNA copies of processed mRNA can be cloned into vectors and maintained as a cDNA library. cDNA gives invaluable insights into both the population of genes being expressed at any given time as well as the mRNA's relative abundance. Now, this EST, which is related with this topic that we discussed now. So, expressed sequence tags, or EST, the partial sequences of cDNAs are often obtained as expressed sequence tags, or EST. In genetics, an expressed sequence tag is a short subsequence of a cDNA sequence. Express sequence tag may be used to identify gene transcripts and are instrumental in gene discovery and in gene sequence determination. ESTs make gene discovery an easier task. The figure shown the mRNA, then the formation of RNA through the reverse transcriptase, then by taking the cDNA through the ribonuclease degradation of RNA through the synthesis of second strand of DNA, and thus double-stranded DNA is formed. Transposition is horizontal gene transfer, that is the transfer of genetic material between organisms other than by vertical gene transfer. Transposons or genetic transposition, a mutation in which a chromosomal segment is transferred to a new position on the same or another chromosome. So, in this class, in this module, we had discussed about the gene expression in prokaryotes and eukaryotes through understanding the genomes, gene structure, GC content, frames, genome frames, etc. The identification of gene expression or gene prediction is the very difficult task. When we get a sequence of organisms of genome, we have two goals, primary goals and secondary goals we can classify. The primary goals when we get a sequence of genome is identification of genes, exon boundaries and splice sites, beginning and end of translation, and the secondary goals include alternative splicing and regulatory elements like promoters. As I mentioned, it is a complicated activity. The only certain way to do this is through the experimental work. But we know it is a time consuming and expensive activity. So computational methods can achieve reasonable accuracy quickly and help direct experimental approaches nowadays. Various algorithms like hidden Markov model, discriminant functions, neural networks, similarity based methods, etc. can give a reasonable accuracy quickly and can support experimental approaches.